Good afternoon, everyone. Thank you for joining this first of two sessions on epigenomics, the first at ABRF. We're quite excited. My name is Claudia Lalancette. I am the Managing Director of the University of Michigan Epigenomics Core, and I will be leading this session. Will start by thanking our sponsor, Watchmaker, for sponsoring this session, and I will invite Jain to come and give us a quick talk on TAPS Plus.
I'm going to start a timer so I'm mindful of everybody's time here. Awesome. Okay, I'm Jen. I'm from Watchmaker Genomics. Today I'll be talking about TAPS Plus, our latest solution for NGS library preparation focused on methylation sequencing. Now, with traditional methylation sequencing approaches like bisulfite sequencing or enzymatic methylation, you protect your 5 methylated cytosines and you are converting all of your unmethylated cytosines to thymines. What this does is it means that over 20% of the bases in your library are being converted, and this collapses base diversity and essentially collapses what was a 4-base genome into a 3-base genome. This has implications for sequence quality, for alignment, and it also limits the utility of these libraries to be specifically for methylation profiling applications. TAPS Plus takes a different approach. We are directly converting your 5 methylated and hydroxymethylated cytosines to thymines, and we are preserving all of your unmethylated cytosines. And this means that only 1 to 2% of your bases are converted, and this maintains sequence complexity and maintains that 4-base genome. Now, this improves sequence quality, it eases alignment, and it also opens the door to leveraging these libraries for genomic profiling applications. Now, you may be familiar with TAPS. It's a method that was published a few years ago. And one thing that I want to convey is that TAPS Plus is not the same chemistry as that published method. We've spent 2 years innovating and developing to ensure robust performance, reproducible performance, improved efficiency and stability, as well as ease of implementation across a wide variety of lab settings. And so, in short, this workflow starts with DNA library prep. We're leveraging the Watchmaker DNA library prep kit for this step to maximize the library conversion of your input template DNA into sequenceable library molecules. We then go into a TET oxidation step using a novel TET enzyme to improve efficiency and performance. From there, you go into a reduction step, and here we're leveraging a novel reducing agent that is compatible with standard lab operations. You can prepare these libraries at the bench. There is no need for a fume hood or specialized equipment. And then lastly, there's library amplification. Now, because this process takes 5 methylated and hydroxymethylated cytosines and converts them to dihydrouracils, we're leveraging an engineered polymerase system that efficiently amplifies DHU-containing templates. Now, end-to-end, this workflow is 6 hours. It's fully automatable on standard liquid handling platforms. And when we compare it to other methods like bisulfite conversion and single-stranded DNA library prep or enzymatic methylation, we see that you can save on the order of 2 hours by using TAPS Plus. It's compatible with 1 to 200 nanograms of high-quality, cell-free, and FFPE-derived DNA, and it's sequencing platform agnostic. Now, at its core, this is a methylation profiling technology, and so what we're showing here is our ability to accurately profile methylation information. On the left-hand side, we're looking at a positive control and showing greater than 98% methylated cytosine conversion efficiency across the full supported input range of the workflow. And on the right-hand side, we're looking at false positive rates, again, across that fully supported input range, and showing a lower false positive rate relative to enzymatic methylation and bisulfite conversion workflows. So takeaway here is you're getting very accurate information with respect to methylation. Now, I mentioned that the fact that we're maintaining all 4 bases opens the door to genomic profiling. So here we're showing the ability of TAPS Plus to access SNV information from libraries, looking at F1 scores. And again, we're comparing across those 3 methylation chemistries I've previously outlined. in addition to adding in a whole genome sequencing control, so an unconverted control. Now, TAPS Plus delivers the highest F1 scores of all of the methylation technologies assessed and is most similar to that unconverted whole genome sequencing control, meaning that, again, you're accessing accurate methylation information and accurate SNV information as well. TAPS Plus is a gentle chemistry. It's non-destructive, non-damaging to your input DNA, and that enables compatibility with low-input cfDNA samples as well as damaged FFPE. Here we're looking at 1 nanogram cfDNA libraries, and just to orient you, the plot here is looking at the number of reads binned by fragment length. First takeaway is that the TAPS Plus library, again, looks very similar to your whole genome sequencing control. And I think very importantly, you're not losing resolution on those shorter library peaks that are so important for applications like fragmentomics. And so this really is enabling in that you can layer in that methylation information in addition to standard fragmentomics profiling. Bisulfite, by comparison, loses resolution of all of those shorter peaks. It also just generally loses information and loses coverage of this sample because it is so damaging in nature. Again, as I mentioned, FFPE compatibility was a big focus for us during development. Here we're looking across 3 independent FFPE samples of qualities ranging from quote-unquote high to low, as assessed by a qPCR assay. On the left-hand side, the takeaway when we're benchmarking relative to enzymatic methylation is much higher library yields coming out of this end-to-end workflow. So you're just generating more library. And on the right-hand side, we're looking at the percentage of usable reads. So of what makes it to your sequencer, how much of that is usable in supporting your biological question. And again, as you can see across this full range, the TAPS Plus chemistry is delivering a higher percentage of usable reads, which is really a function of more reads passing filter and aligning than enzymatic methylation. Again, with respect to methylation profiling accuracy, we see a substantial reduction in the false positive rates across the full range of FFPEs assessed. And the takeaway here is you're getting more accurate methylation information out of these libraries as well. And then lastly, here we're taking a step back and maybe looking at higher level library performance. Looking at something like duplication rate, for example, as a proxy for library complexity or how much unique information is contained within your library. And again, TAPS Plus compares very well with our unconverted whole genome sequencing control and delivers duplication rates much lower than enzymatic methylation. So we're generating libraries that are more complex. We also looked at artifacts, sequence artifacts, and these can be inflated as a function of just the library preparation chemistries that you choose. One such artifact is, chimeric reads. These can be real, they can be really indicative of true structural variants in your library, but oftentimes they're just actually artificially inflated by the chemistry that you choose to prepare your DNA. And so here again, we see that TAPS Plus best aligns with that unconverted control and is much lower in terms of chimeric read rates than enzymatic methylation. So we're giving, essentially improved sequence accuracy of the final libraries. And this also holds true if we look at another type of artifact, like a hairpin artifact, which we're showing on the right-hand side. Again, TAPS Plus here delivers artifact rates on par with that unconverted control and much lower than enzymatic methylation. And so the global takeaway here is that we're able to deliver more complex libraries that contain more information and that also have improved sequence accuracy. So looking holistically across really everything that we've talked about in this session, TAPS Plus delivers excellent conversion efficiency and positive methylation accuracy with low false positive rates, high base diversity that enables applications in genomic variant calling. It's non-damaging to DNA, which really ensures compatibility with clinically relevant samples like cfDNA and FFPE. It's a simple workflow, highly automatable, and can save you in terms of turnaround time and hands-on time. And we didn't really touch on this, but it also can help to reduce computational time and costs because, again, you're aligning a 4-base genome rather than a 3-base genome. I feel like I may be going very fast. I can't tell because I can't see my timer. But if you want maybe a more in-depth paced, deep dive into TAPS Plus, we are having a breakfast workshop on Tuesday morning. It will cover TAPS Plus. It will also cover our upcoming innovations in PCR-free whole genome sequencing with an integrated library normalization solution. Equiplex normalization for standard library normalization applications, and Equinox Prime, which is our upcoming a new NGS library amplification polymerase module. So it's 7:45 in the morning. Boo, that's early. We're all tired. I get it. But mimosas, breakfast will be served. So we welcome you. Hopefully you can make the time to attend. Thank you.
Thank you, Jen. And their booth is 1815. So if you have any questions, don't hesitate to go and see them. That's me. All right, now we come into the core of the session, innovation and assay development in the epigenomics core space. So the way this session was designed is taking into consideration different aspects that can go into innovation and assay development. We can look at it in different ways. There will be evaluation of needs, evaluation of what's available, looking at innovation. If we can innovate in our cores, we're kind of happy, but sometimes the innovation comes from researchers developing platforms that get of interest for what we offer customers. And then the impact of implementation and thinking about how you can implement a novel assay into your core. So that's kind of how this session today was designed. I will go through a kind of model of how I go about assessing or evaluating assays in the epigenomics core that I lead. Also offering another way of maybe engaging more with researchers, especially when you've been there for a while and you kind of know a lot of them. Then Yanxiang will be here for the innovation because he has been one of the co-inventors of the only epigenomic spatial platform that I know about. So he's going to come and give us all the secrets about it and what he's implemented since. And then Abhinav is going to come and briefly bridge on his testing of that platform, but more importantly, give us, from his standpoint, how he was actually able to get high chip to work in a core in a robust way, which I cannot wait to see, because I am not able to do it right now. All right, so the way I approach assay evaluation in the epigenomics core. So this is my personal view, okay? And also you will see that I have a specific case study I'm sharing with you. I do not endorse specific companies. I offer the whole gamut of what's available and depending on researchers' needs and what are their research and their questions. So I want to make that clear. So when you are evaluating, a lot of times we are going to look at what investigators are asking us about, what are their needs, and then sometimes there is a service or an assay that we are not readily, providing for them, and then we want to be mindful of the cost of implementing an assay. We also want to be mindful of serving as many researchers as possible, and there's a balance there that we need to keep. So I see it as adapting to the needs, and then we go and start thinking about how we're going to implement a service or an assay that we've been asked about for a year. But sometimes there are some of those assays that come and we've got a specific gap that we've identified by talking with researchers. And then an assay comes and it looks like it could fill the gap that we are not able to, the service we're not able to provide anymore. And as all of you know, I mean, epigenomics is not new of the last decade. It's been around for a while. But in the last couple of years, there has been an expansion in all of the different assays that are possible to offer, going from cytosine modifications to chromatin organization, even just looking at protein DNA interactions. We are now also entering this field of spatial. And you can also have a combination of all of these different aspects into the same assay. So it becomes a bit difficult to figure out, from my perspective, what it is that would be worth for the investigators and which assay would be most beneficial. So when we're looking at adaptive approaches, a lot of times it's investigator driven or we see trends. I see a lot of my colleagues who run Genomics Core here. I think it's a no-brainer that spatial transcriptomics was going to go in each and every one of your cores because that was a trend. It was a no-brainer to put it. Now, for some assays, like in my core, for Hi-C and cut and run, that came through surveys and demands. So I wasn't having such a big demand for it, but we did a survey and investigators were like, yeah, I really want to have Hi-C. I do ATAC and then I get back that this is just linear. You don't have the 3D interaction between what you say is an enhancer versus a promoter. So we have to start looking at 3D structure. And I'll be honest, cut and run was forced on me by my faculty advisory committee. They said, you need to offer this. And people were struggling doing it in their labs, so I took it on. Then there's other assays that I've implemented, and that's actually a model that blends between the two for single cell methylation, where it's an investigator that reached out to me and said, I want to have an RO1 that differentiates from everybody else. I want to put some data showing that we can do single cell methylation. I know there are papers out there doing it. Can you help me? And I had been wanting to try to implement this because I like driving and pushing the field and being the first one to offer something. So I said, sure, why not? Looked at the papers, we agreed on one, and then that got me thinking about, I've been here for 11 years. There's a lot of investigators that I know that would probably be interested in, looking and testing or providing samples. I run a core, I don't have samples. Investigators have samples. And see if there would be an interest. And that's where I kind of started for some assays, not all of them, taking a bit more of a core-driven or what's called like a proactive approach. When I see an assay and I know some investigators that either I've prepared the samples for them on different assays or methylation or other types of modalities, and then I can reach out to them. Now, adaptive is a lower risk because we know we have, the investigators who are going to be interested in it. We can survey, see if they have, that we can look at what's available. A lot of times there's a couple of kits that we can try. It can be iterative, collaborative as well with the investigators. I will, sometimes say, yeah, I'm happy to test this, get the kit. The development will be on me. I'm not going to charge you for my time because I'm lucky. I do have time on development provided, for my core. And then if it looks good and you want to sequence, you will pay for the sequencing because I don't have the money to pay for that. And in a lot of times, the agreement works well. Now, when we go to a proactive approach, well, there's a bit of higher uncertainty. Yes, that it can be filling a gap or it can be something novel that you think is going to be of interest to certain groups. That being said, it's a bit more uncertain because, yes, if you've got a couple of people interested, it's great, but you want to have something as a service that's sustainable. So you have to be a bit more strategic. And that's where I'm using the Biomodal evoC testing that I did in the core as a model for how I went about with this. So it has to have a scientific fit. People ask me to test some assays. I'm like, no, there's no point. This is not for my core. There's got to be a sample context. I'm very careful about choosing which investigator to approach, what type of samples I will test, depending on the assay or the kit that it's going to be. And you want to have practical considerations. Someone's going to bring me a bioarchive paper. I will have a look at it. I don't necessarily want to start testing it right now, especially if it's in the version 1 of the manuscript. And if it's going to add value, if it's going to bring something that is going to help the researchers, because I run a service. I'm there to help them drive the best research and be successful in answering their questions. So, as I said, I've been running this core. I'm in my 12th year now, and it's kind of interesting because from the beginning, we had a very strong um, group of researchers from different fields in cancer, development biology, environmental epigenomics, and neuroscience that were very interested in having hydroxymethylcytosine measurements, which is not everywhere. I recognize that. And that stems from my founding faculty director, who had shown that in AML, hydroxymethylcytosine was very important. So even when she left, she went to Miami, that stayed. So there's always people asking for hydroxymethylcytosine. It went down a bit. So we were doing 5-HME dip like everybody else. That was about the only thing at the time that was available. And people were happy with that, but then some papers came that you could do hydroxymethylcytosine on a single nucleotide manner, and then the other types of modifications of cytosine, like formylin, carboxyl, and everything. So investigators were calling me and asking, like, I want to do 5-HMC. What do you offer? And I would say 5-HME dip. That's the only one I know that can run. They would hang up on me. So that's not a good feeling for a core director. And so clearly then I was not able to serve them. And there was a couple of instances like that. It was not just one or two and everything. It was like, no, I want single nucleotide resolution. Okay. So OXBS came and we implemented it, had good success, then it faded away. EMC, HEMC came and we implemented them. We still run them. They're great. They're all great. But then one thing that came is that for most of those assays, if investigators want to look at both methylation and hydroxymethylation, then it's two libraries, indirect measurements. And when you tell the investigators it's two libraries, they're like, well, I don't want to pay for two sequencing. I don't want to sequence two libraries to get like one answer. All right. So when the biomodal came, then I got interested in asking, because I was like, clearly I need to give them something that they want to look at both measures and, least sequencing as much as possible. So I had worked with a group since the beginning of the core. It's a group of researchers at the University of Michigan. It's in cancer. And it was actually one of the first projects that I had worked after starting the core, where these hands had produced the whole genome bisulfite sequencing in the 5-HME dip and an HPV head and neck cancer samples. Not only that, they also had genotype, they had viral, because it's HPV integration, they have viral integration, and they've run everything. And that cohort has been going on for, I can't remember when. Now, for my colleagues who've been like me doing sequencing ever since it started in NGS, you will see that these samples ran on the HiSeq 2500, so they're not new. The data is not new. So I was able to reach out to Dr. Sartor and say, hey, I've got this assay where I think it would be great if I was able to use some of the DNA that we use to generate whole genome and 5-HME dip to verify it. I like to not just take the samples that companies have said, like GM12878 or whatever, like, oh yeah, that works great. I like testing with real live core samples. So she said, great. I was like, you'll see the data and like we'll talk about whatever. Of course, because we had done like 5-HME dip at the time, needed 1 microgram of DNA to do the immunoprecipitation, even in a cancer setting. So not all 18 samples had DNA left. And I had a small version of the kit anyway. So I chose 4 HPV minus and 4 HPV plus to drive that test. And I have looked at every aspect, every modalities that the kit claims to, provide. But today, for the context of this, I'm just going to show what we're able to get if in the comparison with whole genome bisulfite and also the 5-HME, because that was the reason why I wanted to test the kit anyway, right? And some other ways that we sometimes will go and look at the data and see if there's anything else we can milk out of it, because there's always something else that you can get out of data. And before I forget, you see this name here. I wouldn't be able to present any of the next slides without Raymond's help. He's my magician in bioinformatics, and he's been a great help in providing, like, we had to break our usual pipeline to run this thing, and he was just awesome. And I'm really grateful to be working with him. So the first thing for the evoC, the first step is that it looks whether a cytosine is modified or not. It does not necessarily differentiate, and you can get that data out. So that's the most comparable to whole genome bisulfite, because whole genome bisulfite does not differentiate between methylation and hydroxymethylation. And I looked at how the two compared. Of course, there's a lot of differences into the chemistry for library preparation. I did not resequence those old ones. I used the old data. So there's a lot of differences in the sequencing and the chemistry for both the sequencing and the library prep. So there is, some partial overlap. It's not as complete. It comes a lot from evoC being enzymatic based versus whole genome being chemically based, because as we all know, it's very harsh. However, I wanted to look for those regions that do overlap between the two, if there was good concordance between the signal. This is coming after comparison of HPV, the differential methylation of HPV to plus to minus, because I processed both side by side and then I looked at the comparison. I thought it would be an easier comparison to do. And we do have good concordance. There's this group of dots here that look completely different. I haven't looked into those ones yet. I'll be interested in seeing. So it did look like it was giving relatively for the regions that they shared, different, like similar examples. I also had a look at it for hydroxymethyl cytosine. I don't know why the colors disappeared, but that's okay. And in this case, well, it's an immunoprecipitation versus a conversion in single nucleotide resolution. So I did not, do like the reanalysis and everything. So I went like sample by sample. I also did not have the original files to rerun all of the 5-HME dip. The researchers can't find them. I don't know why. So I'm still hoping they will find them soon. But overall, for this, all of the regions from the 5-HME dip that were pulled down when we did the libraries are covered by the evoC, which is something that was interesting. Now, the next thing we asked was, okay, so we get these differentially methylated regions, we get these differentially hydroxymethylated regions, and the thing about that assay is that they all come from the same read. So you always have the same read, and then it measures modification, and then it differentiates whether a cytosine was methylated or hydroxymethylated. So then we started looking at, is there any overlap between the differentiated region and with the significant regions that from the analysis for the evoC modalities, we were actually seeing some regions that are shared between the two. And because we're doing differential between two groups, so it means that in one group, methylation is higher, whereas in another one, it's hydroxymethyl cytosine that is higher, which we rarely are able to do with any other assay right now, except if you are actually doing, I mean, it can be done, but with that amount of data, unless you go on an array where everything is being probed, for exactly the same at the same probes, it becomes a bit more challenging, and in some aspects, you need to have a lot of sequencing to be able to do it. So, and that's where Raymond's genius comes. We said, well, okay, so that's if we're only looking at regions that coming from, we looked at differential methylation or hydroxymethylation. So those are where the significance is there. Regions that we're not picking up, but there is some sort of a shift or a switch between the two conditions. And there had actually been a paper that came out where they were doing that type of analysis using the Illumina arrays, the methylation arrays, where they were looking at it. So I gave that to Raymond, and Raymond just implemented that in our pipeline. Wasn't easy, but he got it done. And what we're looking at here now is those regions where in HPV plus, if it's red, they are predominantly significantly methylated, and then if it's, then they go down the negative here. So if they're positive, they're highly methylated in that condition. If it goes below 0, then there's a shift towards the other mark. So we're able now to look at shifts between conditions where in one group it was predominantly methylated or hydroxymethylated, and then in the other group it's completely different. And that can also give insight into some shifts in pathways and different other questions that were very challenging to address before. So I've tested all the other things, but for the sake of time, I'm not going to go over them. So that's kind of showing how sometimes you can leverage, because I think everybody, doesn't matter, you don't need to be in epigenomics, you can leverage long-term collaborations, and you know your investigators you've been working with, you do the consultations, you talk with them, and you know sometimes they have not as well characterized, but they will work. You can work with them, and they will be more than happy to do it. And so specifically for this assay, yes, it does measure both modalities. And I think also the nice thing is that as a core, we're able to develop an added value to what the kit offers. And that is, shown by implementing this change in methylation, dynamic change that we can see now added to the pipeline. And lastly, Raymond, thank you. I want to thank my sequencing core because I wouldn't have had that data without them. And they were pretty fast when we had it. And of course, Dr. Sartor, who allowed me to use our samples. Thank you. And now I will welcome Yanxiang from University of Pennsylvania to come and talk to us about the spatial. We'll just go all, and then if you have questions, we can answer them at the end. Sorry. I was too stressed to specify that before.
Hello, everyone. I'm Yanxiang from University of Pennsylvania. So today I'd like to introduce several spatial technologies we developed that can profile different type of molecules within the tissue section. So we call this spatial multiomics. So first of all, cells are building blocks of life, and the cell states are determined both by the intrinsic and extrinsic factors. Intrinsically, cells are modulated by different omic layers, including the genome, epigenome, transcriptome, proteome, and metabolome. On the other hand, cells are affected by their neighbors through the cell-cell interactions. Therefore, we need to develop a technology that can profile different layers of omics within the spatial context so that to better understand the cellular states. So traditionally, bulk sequencing are widely used. However, we need to mix different type of cells as a bulk input, where we only get the average profiles from all cells which will review the heterogeneity among different cell populations. So to address this challenge, single-cell technologies are being developed, which can compartmentalize individual cells using either microfluidic devices or microwells to get distinct profiles from different cell types, so that we can get cellular states from individual cells to review the heterogeneity among different cell populations. This technology is very successful, and we generate a lot of spatial single cell multi-omics profiles from different tissue types and species. However, as we can see, this technology has some limitations. For example, the microfluidic devices have a relatively low capture rate, which means that we will lose a lot of cells, and also the dissociation process may introduce some artifact. And some cells are also very fragile, and the dissociation process will lose those cell populations. More importantly, we will lose the spatial information, which is important to understand the biological process within the tissue sections. So to address these challenges, spatial omics, or especially spatial transcriptome, have emerged as a powerful tool to profile biomolecules without dissociating the tissue section. In generally speaking, there are two types of spatial omics technologies. One is the imaging-based methods. The other one is NGS-based methods. So for each imaging-based method, most of them are using the in situ hybridization or in situ sequencing, where you can get very high spatial resolution, can go down to subcellular resolution, and you can get this kind of like a single molecule signals from within the individual cells. However, we will need to design the specific probes to target the molecules of interest, which will limit the coverage of the gene you profile. On the other hand, NGS-based methods can achieve genome-wide profiling, which is an unbiased method. However, this technology will have limited spatial resolution, which is always limited to this spot-like resolution. With the spatial information, we can do some kind of a unique data analysis. For example, we can analyze the spatial neighborhoods, also at the tissue level, and also we can study the cell-cell interactions through the ligand receptor interaction study. And finally, at the molecular level, we can check the spatially variable genes within the tissue section. So, for example, if we want to study the cancer with the spatial information, we can look at how do cancer subclones grow in 3D tissue or 3D microenvironment. And at the molecular level, we can look at how do cancer subclones interact with each other and also with the tumor microenvironment. Finally, at the molecular level, we can look at how does the tumor microenvironment influence the growth of the cancer subclones at the molecular level. So currently, most of the commercial technologies, spatial technologies, are focusing on the spatial transcriptomics, which is a gene expression. As mentioned earlier, it's not a kind of full picture of the cellular states. So recently, we pushed this field a little bit further that can co-profile or joint profiles gene expression and the panel proteins within the same tissue section, and we call this a DB-seq. So the idea is very simple, as DNA can hybridize with the complementary basis pairs, for example, the A2T or C2G. So we can design specific probes to bind or target the molecules of interest to do the spatial barcoding. For example, if we are interested at the messenger RNAs, since it has the poly A tail, we can design the oligos with poly T tails together with the spatial barcodes to capture these messenger RNAs and barcode the messenger RNAs at the same time. And after we sequencing these molecules, we can reconstruct their spatial location. So to deliver the spatial barcodes into the tissue, we design these microfluidic devices. We have two sets of microfluidic devices. One is flowing horizontally, the other one flows vertically. So here is a real picture of the device. We have, for example, 50 inlet and 50 outlet. We add the spatial barcode solution to each inlet and then use the vacuum to introduce the spatial barcodes on the top of tissue section to deliver the spatial barcode into the tissue. So if we want to capture the messenger RNA, as I mentioned earlier, we can use the polyT-tailed DNA oligo with the spatial barcode A to hybridize with the messenger RNA. And to capture the protein or surface protein, we use the ADT, which is oligo tagged antibodies to barcode the spatial location of the surface protein. So for the second round, we will allocate the barcode B to kind of encode or barcode another axis of the spatial location. So afterwards, we will have this cross flow pattern on the tissue section. So at each intersection, we will have a unique combination of the spatial barcode A and B, so that we can distinguish their spatial location afterwards. So then we took the image of the tissue and then lysed the tissue to kind of connect the barcoded cDNA and the ADTs and then send for sequencing. Finally, we can computationally reconstruct the spatial profiles of either messenger RNA or proteins. We tested this technology on the embryo at the very beginning with different spatial resolution and so on the mouse embryo. So at 15 micron resolution, we can resolve the major tissue architecture in the embryo, for example, the forebrain, the heart region, and the spinal cord, and so on. And then we can further increase the spatial resolution to zoom in to look at the embryonic brain region, where we can resolve this microvascular network. Finally, we can further increase the spatial resolution to 10 micron, which is close to the single cell resolution, and focus on the kind of embryonic eye area, where we reviewed this single layer of melanocytes in the optical vesicle. As I mentioned at the very beginning, so the cellular states are modulated by different omic layers, and so profiling the epigenome in addition to the transcriptome and the protein is also very important, because the epigenome is kind of like a software is controlling the gene expression and the cellular development trajectory. And also the non-coding DNA represent 98% of our genome sequence, which are associated with many disease risks. So we take advantage of also use the DBSeq platform strategy to barcode the different type of molecules, including the histone modifications, the accessible chromatin, to do the kind of spatial epigenome profiling. And we developed the spatial Cut&Tag to look at histone modifications, and the spatial ATAC-seq to look at the spatial chromatin accessibility. And most recently, we achieved spatial co-profiling of the epigenome and the transcriptome within the same tissue section. So, most recently, we developed another spatial epigenome profiling technology that can look at DNA methylation within the tissue section together with the gene expression. We call this platform as a spatial DMT. So here only shows the workflow for the spatial DNA methylation profile, how to profile the spatial DNA methylation. So first of all, we use the 0.1 N NCL to treat the kind of a tissue to remove the nucleosome, and then use the TM5 to cut the whole genome into fragments, and also insert the DNA adapters. Then we use the similar strategy with the DBSeq to use the microfluidic device to barcode, to do the 2 rounds of barcoding within the tissue section. And then afterwards, we retrieve the barcoded DNA and then perform the EMC and to convert the unmethylated C to T, so that we can differentiate methylated C and unmethylated C afterwards. So we similarly we applied the spatial DMT on the embryo at the very beginning. So here we can see that both demethylation and the gene expression can resolve the major tissue types within the embryo. And interestingly, we also found some demethylation can resolve some subtle region-specific epigenomic variations that transcriptome cannot resolve. For example, when you look at this kind of light blue cluster, the gene expression cannot differentiate these two regions. They just the gene expression only identify this region as a one cluster. However, when you look at the demethylation, it can differentiate these two regions as two separate clusters. So we are wondering whether demethylation can identify more features or to differentiate these two clusters. And indeed, we found that we identified a lot of differentiated methylated regions between the red cluster and the blue cluster, and also they enriched a lot of motif regions related to the biological process. However, when we compared the gene expression between these two regions, we only see very few differential expression genes, which means that when you compare these two regions, there's a more difference in the epigenetic level compared to the transcriptome level, which may be due to the epigenetic priming. So then we increased the spatial resolution and tested this technology on the mouse embryo. Again, the demethylation, the gene expression, and also we integrated both modalities, which can further refine the spatial clustering of this mouse brain regions, which specifically the hippocampal region. At the beginning, the demethylation and gene expression cannot resolve the superficial cortical layer and the deeper cortical layer in the mouse brain region, but after we integrate both modalities, we can differentiate this like the superficial cortical layer and the deeper cortical layer. And with the with the gene expression profile, we can do the cell type deconvolution computationally by using the single cell reference data. And again, by using this technology kind of computational methods, we can further refine the spatial resolution or cell type in the tissue section. For example, different excitatory neurons enriched with different cortical layers, and also the CA regions and dental gyrus. So as we may know, in addition to MCG, MCA also is very abundant in the neuron cell. So here with our technology, we can look at the MCG, MCA, and together with the gene expression to study how they are correlated with each other. So we used this scatter plot. The y-axis is CpG methylation level of fold change. The y-axis is CPA methylation fold change, and the color is corresponding to the gene expression fold change. So we found that their correlations are very complicated. Some of them are positively correlated, some are not. So for example, for the PROX1, which is highly expressed in the dental gyrus region, showed negative correlation between the MCG and MCA and the gene expression. However, for the NTRK3 gene, which is showed the high expression in the CA1 and CA2 regions, only negatively correlated with MCG, but not MCA. And then COX1 gene showed like lower expression in this CA1 and CA2 is negatively correlated with the MCA signal, but not MCG. As we can see, this kind of three layers of molecules have very complicated regulation relationship. So I think, as mentioned at the very beginning, the cellular states are modulated by different layers of omics. So to better understand the cellular states, it would be better to look at as many as molecule layers at the same time within the same tissue section. So that's why we recently developed a new spatial technology called the spatial MOC-seq, which can map chromatin features, transcriptome, and the panel proteins together within the tissue section. So the chromatin features including the chromatin accessibility and 2 types of different histone modifications. So in total, we can profile 5 different modalities within the same tissue section. So we take advantages of the nanobody TM5, which has a species-specific binding characteristics, and also therefore we can insert the barcode adapters with different to barcode different modalities. So we first tested again on the embryo as well. So you can see that we co-profiled chromatin accessibility, K4 trimethyl, K27 trimethyl, and the gene expression. And all modalities can resolve the major tissue architecture within the mouse embryo. And then we checked individual gene chromatin accessibility, for example, the E2F2, which showed very high chromatin accessibility in the liver region. And however, we didn't see the high gene expression in the same region. So if we only have the information, or if we just co-profile the chromatin accessibility with the gene expression, we probably can not kind of explain what is going on here. So, however, we have another 2 layers of histone modifications. One is the K4 trimethyl, and the other one is K27 trimethyl. And you can see here both signals are showed very high signals, which means that at this gene loci, probably there's a kind of bivalent status at this gene loci. Then we increase the spatial resolution to profile the mouse brain, and using the K27 trimethyl, K27 acetylation, and also gene expression. Again, different modalities can resolve the major brain regions, and after integration, we can further enhance the spatial resolution after spatial clustering. And interestingly, when we look at the dental gyrus region, we can further kind of differentiate the subcellular, subgranular cellular zone and kind of dental gyrus region. These 2 regions have very specific regulation patterns. For example, for this IGF-BP1 gene, which is a marker for the SCG region, seems correlated with a K27 acetylation, but not K27 trimethyl, because when you look at the gene loci, there's a huge difference or significant difference between the K27 of the K27 acetyl signal between the SCG and the CG regions, but you didn't see the huge difference in the K27 trimethyl. However, for the POX1 gene, which is a marker for the SCG area, which seems correlated with both signals. For example, the K27 acetyl showed increase in the SCG together with a decrease of trimethyl of this at this gene loci. Finally, we demonstrate this technology on the mouse brain with full 5 modalities, including the chromatin accessibility, K27 trimethyl, K27 acetylation, gene expression, and the panel proteins. And again, all 5 modalities can resolve the different brain regions. And we also check some marker genes like CD140A, which is enriched in the corpus callosum, and corresponding to the high gene expression and the K27 acetylation and the ATAC. But interestingly, when you look at the K27 trimethyl, it also showed very high high activity or high signal. However, K27 trimethyl is a repressive loci, which should different or anticorrelate with this protein signal. However, then we zoom in to look at the genome browser, what's happening here. And we found that the gene expression at this gene loci is highly enriched for the longest isoform of this gene, and the K27 trimethyl signal is low at the promoter of this at this gene loci, but we see high signals of K27 trimethyl for other isoforms, which indicating the selective epigenetic separation. So up to now, all the spatial technologies we developed are only compatible with the fresh frozen samples, which is a huge limitation because clinically, most of the samples are stored as FAP samples. So to address this challenge, we recently developed a new technology called spatial FAP ATAC, which can be compatible with the FAP samples to look at the chromatin accessibility. So we did extensive optical, protocol optimization. Here it's a summary of the different protocol conditions, but we tested like hundreds of different conditions that achieve this kind of good data quality. Generally speaking, we tested the targeted retrieval temperature, protein K concentration, protein K incubation time. As we can see that we played with different conditions, and we see the ATAC signals become better and better. And we also test a different targeted retrieval buffer, and we also see improvement of the unique fragments from these FAP samples. So we first kind of test the feasibility of this protocol on the mouse brain as well, and the spatial clustering looks very good, which can differentiate the cerebellum, different regions of the cerebellum and the hippocampal region. And then we checked the signals or marker gene activities between two biological replicates, which showed a very high spatial kind of specificity, and also a high, and also they're reproducible. Of course, we want to compare with fresh frozen samples, see how it goes. And again, they are correlated with FAP samples, and the fresh frozen samples are correlated with well. And I want to point out that when we compare this gene expression, spatial gene patterns between the FAP samples and the FF samples, we see FAP samples showed more distinct spatial patterns compared to the FF samples, indicating that FAP samples it's better good, maybe it's better at preserve the tissue morphology compared to the fresh frozen samples. Then we move on to test on the human samples as well, and we test on the human cerebellum and also human thymus, which also showed very good spatial patterns. And we further increased the spatial resolution from 50 micron to 10 micron using the Atlas Omics flow gel, which is showed very good, more refined spatial patterns in the human thymus. Finally, we tested on the kind of the human melanoma samples, and we can resolve the distinct spatial regions in the melanoma. For example, the non-tumor clusters and the tumor clusters. Within the tumor clusters, we can further review the heterogeneity among different tumor regions. And finally, we checked the marker gene activities among different regions, and including the PD-CD1, BRAF, and also NRAS, and so on, which showed very distinct spatial patterns. Finally, we just want to mention that we share all this data and also how the code and also protocols online. And with that, I would like to thank all my lab members, collaborators, and funding sources. Thanks for your attention. Happy to take any questions afterwards. Thank you.
Last but not least, I'm losing my voice. I won't be able to karaoke. Abhinav from ND Anderson.
Thank you, Claudia, and thank you all for staying, and great presentation so far. So I'm going to talk about some of the innovations that we are doing, because that's the theme of the conference and also this session. When I get a chance to public engagement, I would like to show a little bit about what Epigenomics Core at MD Anderson offers. So this is our mission statement, that we basically are here to facilitate both basic and translational epigenomic research, because we are biggest cancer center. We have a lot of colleagues who actually have access to clinical samples. They would like to understand epigenomic profiling in that, so we kind of support them. And as you might have heard by that time, I mean, if you are in this room, you know that our DNA fiber is not linear. There is a lot of information up to the chromatin and 3D state. And how do we resolve it? So we have the core is actually divided into two sections. I lead the chromatin analysis part, where we provide all these different expertise and assays to resolve histone modifications, DNA protein interactions, chromatin accessibility, and 3D genome profiling, which a major part of my conversation today would be that how we optimized our assay. I'm supported by these two talented technicians. The staff in the lab, we are small, but not small in the output. And we recently have onboarded a bioinformatics support faculty. She leads a bioinformatics group and a lot of analysis pipeline that we deal with all these omics assays. We also have DNA methylation, which is led by my colleague, Marcos Estasio. So they have all kinds of bulk and in cfDNA, the methylation analysis. So I'm not going to talk about that part. So this is a little bit of the project workflow that anybody enters at the core. We basically, as a scientist ourselves, a lot of these folks, they want to do epigenomics, but they don't know where to start. So that's our job to advise them on the best assay, what samples you would need, what kind of sample preparation needs to be done. And then at the core, in the red box, you would see basically those are the assay-specific pipeline workflows that we have. But one thing which I would like to highlight is the QC, that's the keyword you would see a lot in my presentation. We do a lot of QC because they depend on us, and they are investing their funds into us. So we like to deliver as best as possible. So once we are not a sequencing core, the libraries goes to the NGS core, data goes to investigator, and then I can talk about myself. I sit in a lot of collaboration meetings and a lot of lab meetings with investigators across MD Anderson and beyond, and we are involved in the data output and how to design manuscript, how to interpret those data, and even for their grants. This is our portfolio for the chromatin analysis. We started a few years ago just doing chip. We have now high throughput version of chips. We have high throughput version of cut and run. We started doing a lot of ATAC long time ago, but then considering we are in a clinical setting, now there is a lot of interest about this cell-free DNA, so fragmentomics, as we heard earlier. So we have established protocols now to do CF-ChIP-seq, and this is from plasma. So a lot of clinical colleagues now, they can follow their patients in a very non-invasive manner. And we are developing the spatial ATAC-seq from the technology that Yanxiang just mentioned. And we recently actually launched high-C, high-chip assays. So that is what Claudia asked me to focus on. How did we optimize in a core setting? So I'm going to show that. So as you see, the punchline down there, every check counts, every outcome matters. That's why we like to do a lot of QC in our workflow. This is what we have achieved. We have handled tissues, cells, all kinds, primary, PBMCs, organoids, mouse, patient-derived xenograft, and we have also special cases where my colleagues, they have tissues on their slides, and they had to scrape that tissue, and we could do ChIP-seq out of those. Same times, tissue cells scraped off the colon, and we have actually over 100 repertoire of more than 100 antibodies we have done chip and cut and run for, and we always reinvent our protocols to make it more refiner, to be able to do from less material and get more efficient output. And we always are developing behind the scenes new assays. So to highlight it here are ATAC-seq and high-C, high-chip that I'm going to talk a little bit about. So not to follow up on the Yanxiang, I'm just going to show a couple of slides how we partnered with Atlas Axomics to use their platform to try to validate, to optimize spatial ATAC-seq in our facility, and also the 3D genome analysis assays. So as Yanxiang mentioned, now this with the ATAC-seq, spatial ATAC-seq, you can actually map chromatin accessibility or gene transcription on the tissue samples. And he also described technology, so made my work short, is basically using this dual par coding. I think what Atlas Exomics did in last few years was instead of using that big vacuum manifold, which we would have to invest money to buy that instrument, they have this clamp system now where you can just stamp those barcodes which are gel infused on the tissue. So this made life easier. So we kind of partnered with one of our colleagues at Cell Therapy Institute, Dr. Rizwani's team, and I'm going to just show you very simple, crude version of these images we took. So brain, as Yanxiang showed, they use positive control for Atlas Exomics, but we had this test sample from Dr. Rizwani's team. They were trying to find out if NK cells would invade these tissues. So, and these lines are just showing that, I'm just showing that how that clamping system made life easier, because by following those lines on the microscope, we could see that all those barcodes got properly stamped into the tissue section. But as I mentioned, we do a lot of QCs. Yes, we made libraries, but one of the ways to look at the library quality before we could invest in sequencing is to look at the fragment analysis. But what we also do is we run a lot of qPCRs. A lot of our QCs is done by qPCR. So here we have chosen primers which we use for our bulk ATAC-seq analysis to profile the enrichment of the open chromatin region. So we could actually see those libraries are actually capturing open chromatin region. Then we actually send it for sequencing, because without this, we would actually not send for sequencing, because we don't want to invest investigators' money or our own money on something which we don't know will work or not. So this is post-NGS, the library profile. You can see it looks pretty good. And then when we profile those open chromatin regions, you can see there is very high specificity on brain-specific versus pancreas-specific samples. So the experiment worked. And now we are going to be partnering again with Atlas Exomics, and we have a lot of interest now based on this initial optimization, which I've shown to my colleagues at MD Anderson. So we are going to do a lot of these, and I think the latest one, which Yanxiang showed, that would be more interesting to do multi-omics actually, rather than just doing one. So that's about ATAC. I'm going to spend most of my time about understanding what information chromatin has. So as we might have heard, chromatin is divided into compartments, into topologically associated domain, TADs, loops. So you can do these chromosome conformation capture assays, and based on the computational profiles, you can break that chromosome into different resolution to call the loops, the TADs, or compartments itself, or you can look at a whole at whole chromosome level. So the technology, the workflow is pretty straightforward. You cross-link the cells, you digest or fragment or use MNAs to break the chromatin, and then you use biotin to fill the ends, and then you sonicate that DNA, which is biotinylated, ligated, and from that sheared chromatin, you can either directly make the library, and that would be a high-C experiment, which would be to detect chromatin interactions on a genome-wide scale, or you can use an antibody to pull down that ligated material based with a specific antibody that you want to use, and then you do the library prep and amplification, and that would be high-ChIP, which is a target protein-associated chromatin interactions. So we basically are very interested, as a scientist myself. Claudia mentioned about proactive approach. So this was very proactive approach. We wanted to have these assays available for our community and once we have it, then they get to use it. So we partnered with Arima Genomics. Brenda and Andy, they have a very extensive, and thankful for their support. So this is very simple workflow. You have the cross-linked chromatin, you break it up with the enzymes, you ligate it with the biotin and fill in, and you shear and purify the DNA. So I'm going to now talk more about QC, because as a core facility, these are very expensive experiments, and unless we do all these QCs, we are not sure where we are heading. So one of the QC steps is to look at the biotinylation efficiency. So we basically, before even making the library, we wanted to make sure, and you can see here, out of 3, 1 sample failed that biotinylation. So that itself tells us it's not worth proceeding further with that experiment. Then after sharing, before you make the libraries, we do another QC to just confirm whether we are getting the fragment size in the range that we are expected to have before we make the library. And finally, once we make the library, then we look at the library QC again before it is sent out for sequencing, just not to spend everyone's money. I'm going to give you an example from an active research project that I have my own lab. We work on link RNA. We depleted this link RNA. We have this whole story, but we use this cell line as an example when we were establishing this assay. You can see this contract matrix after high C. On the bottom panel, you can see there is a little bit differential chromatin contacts when you deplete this link RNA. We wanted to look at it further, and when you look at this loop file, now you can see on the top it's control cells. Here is the link RNA depleted cells, and there is new loop forming. So this example we saw across the genome at different locations. And what we can do is we can actually use the CTCF and other histone modification ChIP-seq data or cut-and-run data to map now because CTCF is supposed to be anchor for some of these loops, and now we could show that there is CTCF anchors on those loops. So those new established loop formed was not just a fluke, it is actually happening in these cells. So we looked at some of those gene of interest, and these are estrogen-induced genes if someone works on breast cancer and estrogen response. And what we saw that whenever we depleted this link RNA in these 3 genes, we are seeing these new loops forming, and all those loops are at CTCF binding sites, and they're also connecting enhancers to these promoters to cause activation of these genes when you knock down this link RNA. And what happened is this matched to some of the RNA-seq data that we already had in our lab. So we had this data to show that when you deplete this link RNA, all these gene expression goes up. But now with this high C data, now we can actually conclude, and we can really get the biology that those new contacts which are happening in the genome is causing the expression of these genes, and we also proved it by having the ERL for ChIP-seq data. So that was high C. We learned a lot of new biology by establishing the high C pipeline in our core. And next I'm going to talk about high ChIP, which actually is the beast around here. So this is an image from Dr. Bingrang's group, and I have nothing against kits, but I'll tell you one thing why we actually deviated from kit to do this experiment. It's very expensive. We talk like $800 per sample to just the reagents when they arrive to the core, and now you add the core facility charges, labor cost, and the wastage, instrument use, and all that. So an investigator will end up paying almost like $1,500 to $2,000 just to run one sample for high ChIP. We didn't want it to do that, and I'm a scientist myself. I was like, okay, there are published protocols. Why can't we use these protocols and add some QC steps so to make it cheaper and more controllable? The other thing the reason we wanted to optimize the whole protocol in our lab was because if you use Dovetail, Arima, or any commercial kits, those will work definitely, but you have to follow those antibodies, those dilutions. We have years of experience doing ChIPs. We have established all these antibodies. I didn't want it to reinvent the wheel to try a new antibody and see if those ChIPs will work or not. So what we did now, we adapted this protocol. We could use our old ChIP protocol into it. So the protocol is pretty simple. Same thing. You cross-link the chromatin, you biotinylate, you ligate. Now you share, but you add a ChIP step at the end. So now we actually put all these QC on there. So every step we are QCing so that before we do the final ChIP experiment or NGS, we can make our go, no-go decision. I'm going to walk you through a couple of examples. It might look like your molecular biology 101, but these are 4 reactions for 2 different cell types. Here we partnered with my friend Goswami, Sangeeta Goswami at Ellison Institute. So this is the chromatin you get. When you digest, you should see a smear. When you ligate, that smear should go back to bigger chromatin pieces. and now you do the chromatin shearing. So this looks pretty good. But now on this sample, now we have issues. We are not getting digestion, so we are not getting shearing, and subsequently the chip is not going to work. High chip is going to fail from these samples. So the optimization is needed at every step, the digestion step, the ligation step, and the shearing step. So in this case now, for the same cell type, we spent quite a bit of time to optimize the digestion. Now you can see shearing happening. We optimize ligation. Now we have to optimize shearing conditions. So, and after ligation, we also did the biotinylation efficiency one more time before you do the high chip, because your whole enrichment is based on the biotinylation of the DNA. So we wanted to make sure biotinylation is happening. But now if you guys have purchased biotinylated nucleotide, you know how expensive that is. So if I were to do this shearing experiment with using the biotinylated nucleotide, it would cost a lot of money just to run this experiment. So what we did is we just replaced the biotinylated nucleotide with an unbiotinylated nucleotide just to optimize this step. So this is just it was a very good learning experience for my technician and for us as well. But now you can see see the digestion step, ligation step, and different time points of digestion. We run on agarose gel, we could run on a tape, get a better quantitation, and boom, here we are. So we now know that for these type of cell lines, this is the perfect condition we need to do after following all those steps before you do the chip of a high chip assay. So now we got good shearing. So we have all the system optimized perfectly to do high chip assay in our lab with each reagent purchased individually. But now you notice there are 4 different QC steps. There are a lot of actually record keeping. So we have developed these huge worksheets where each module is a go, no-go decision. We track all the chromatin. We don't want to lose the chromatin at any step because you have so many. So, but once you reach at this final step, that is where you can do high chip. If my technician showing me the data pass, and you have this much chromatin, then we do the high chip. I'm going to just give you one example. We did high chip for few antibodies, but here is for K27 acetylation mark, which is actually used to understand the promoter enhancer contract, active promoter active enhancer contract. We did in 2 different cell lines, MCF7, and this is a primary murine T cells. And again, here you can see the library size, fragmentation looks pretty good. So we would not send it for sequencing again before that. And I said earlier, we do a lot of qPCR before we send things for sequencing. So we, based on our own experience with chips, with hundreds of antibodies, we know where to look for the positive enrichment of those antibodies. So based on this, you can see the negative. There is no enrichment of K27 acetylation in both of these human and mouse samples. Then we send it for sequencing. So just to give one quick example, so this is a K27 acetylation high chip. These are the loop files. You are supposed to see these loops. You can see the loop happening from here to here. We also have the chip-seq data for a couple of histone marks like K27 acetylation and K4 trimethylation from the same cell lines. The reason they are mapped here is to differentiate between enhancer and promoter of those genes. So now if I zoom in on this location, you can see this particular promoter of this gene is now mapping to this region, which is very heavy in K27 acetylation. So now by using this experiment, we have mapped a new enhancer for that particular gene in this cell type. Here is another example for BCL11 gene. There are a bunch of enhancers mapping, but when we zoom out, we can see there is very heavy K27 acetylation chip there, but no K4ME3, so that means that is the established enhancer for that particular gene. There is a lot more information you can get. I have put them in the right side. So you can see this gene has multiple enhancers. Then you can map the transcriptional hubs using that data, and also multiple loops, like all the transcriptional active promoter-promoter contacts and promoter-enhancer contacts. So I hope I showed you that we could actually optimize this high chip protocol in our lab, although nothing against the kit which is available and very successful out there. So, but this is actually a lot of work done by my technicians. So this is just to close it out, this is the vision of our EpiCore at MD Anderson. We call in short EpiCore. So we have assays pretty much for every dynamic of chromatin from a single base resolution to the three-dimensional. We have invested a lot of our effort, and we are actively doing a lot of from the liquid biopsies, and this is something we are developing, and I hope by next year we would have more data to show you. This is the impact we are creating. We are not just serving MD Anderson or medical center. We have a relationship with a lot of universities across the United States, even across Atlantic we have worked. And in last couple of years, you can see the impactful science which is coming out of our core, and all those asterisks are where we are listed as active co-authors, because we are not just involved in running the assays, we were involved in running their science as well, to design the science and design the program and the proposals as well. Short team, on the left side is the chromatin group, on the right side is the DNA methylation group. We are supported by bioinformatics with Dr. Yuval Lui. We are very fortunate to have a lot of institutional support, both in money and administrative support, operational support. We are also partially funded by CCSG. Now we have advisory committee, and of course, for those assays we optimize, we have leveraged a lot of technical support for Exomics and Andy from Epigenomics, and so. And with that, I hope I satisfy you that high chip is not a beast. Let's do it. So thank you so much. Thank you, Claudia.
We're all very passionate, so we're right on time for this session. I think if you have questions, just come and see us, or we can take a couple. I see people hovering around in the back, I think, for the next session. Sorry about that. My fault. That was too fast. Yeah, it's 4. Yeah, no, it's not bad.
Yeah.
Great job.